Good afternoon. Today is Wednesday, November 18th, 2020. Here in western Massachusetts, it's getting a little colder now. The fall is definitely in the air and the leaves are on the ground. Snow can't be too far away. I'm Stephen Rich, and today we're going to present to you our 11th Tick Talk of the year. We do it once a month. Today I have my guest, Dr. Sam Telford. This was a pre-recorded episode from a couple weeks ago, so you hear Sam reference the snow that was on the ground that day that we spoke. It's a real pleasure to introduce Sam. So Sam is the person that got me into ticks and tick-borne research many years ago, while he was then a research fellow at Harvard University and recruited me and mentored me at the laboratory of Andrew Spielman at the Harvard School of Public Health all those years ago. So Sam has always been a great friend and a great supporter, a great colleague. I've learned a lot from him, and always grateful for his generosity of spirit and of his time and the knowledge which he has, which is near encyclopedic. Today, Sam's going to talk about one of many subjects he knows a lot about, and that is Babesia. And in a part of our conversation that we taped, Sam talked a little about his career and how he got to where he was. I've sliced that out of today's presentation, but I'm going to link it in the follow-up video. So those of you that are curious about Sam, or maybe aspiring to be a great scientist like Sam someday, you can go on and figure out how he got where he was, because it's really a fine story. He's an extremely prolific author and scientist, and a valued colleague. And so I hope you enjoy the conversation that we taped here with Sam today. Hey, Sam.
Hello.
How are you?
Good. Shocked at the snow.
So do you want to switch over to some slides?
Sure.
So what do people need to know about Babesia?
Well, they need to know that they're parasitic protozoa. So things like Lyme disease, ehrlichiosis or anaplasmosis, Borrelia miyamotoi disease, those are all caused by bacteria. Parasitic protozoa are more complex organisms. Here we have a phylogenetic tree or a tree of life, where you see the fungi and the complex organisms, the molds, the plants, and this group called the alveolata, which include things like malaria parasites as well as their close relative, the Babesia parasites. The Babesia are the second most common protozoan to be found in blood in animals anywhere in the world, blood of mammals anywhere in the world, and they are transmitted between those animals by ticks. They are required to transmit the infection, to keep the infection going from generation to generation, with two known exceptions. We do know that it can be inherited, but in some instances from mother to offspring. And of course, because it's in blood, any blood-to-blood contact can transmit it. So fighting among monkeys, there have been primate colonies where there are no ticks, but plenty of infected animals. And it was quickly found out that was due to fighting and contamination between the blood contamination between them. There are great number of species, at least 100, if not more. But there are many different kinds of mammals and birds, and many different kinds of ticks. And they all locally adapt and eventually diverge. But people need to know that these parasites, these protozoa, are parasitic. They're like malaria. They are obligately living within red blood cells and other tissues, that they eat the innards of the red blood cells. They take the fats and the organelles and reproduce. And if their reproduction is not controlled, they can kill the host by destroying too many of the red blood cells. And so it's all a matter of being able to control the infection. And most of the animals in which you find Babesias, any of the species of Babesias, it's rare to find symptomatic animals or those showing signs of clinical illness. They have come to a balance. The parasite replicates for a while, immunity kicks in, and then keeps the parasite at a low level, while the low level of parasites keeps producing immunity. It's a concept that came about from the very first studies of babesiosis. First description of a vector that is an arthropod that was required to keep an infection going between animals was in the instance of Texas cattle fever in the 1890s by Theobald Smith and Fred Kilbourne, who investigated this endemic condition where cattle would develop a hemolytic disease, their red blood cells would be destroyed, and to the point that their urine was red. They're called red water. This was obviously not a good thing. The Texas cattlemen and others there were very upset about having all these losses due to something, some infectious process, and the USDA sent Smith and Kilbourne to Texas to investigate. And you know, the farmers aren't, they were pretty clever. They knew that it was usually when they went to pastures that had previously been infested by cattle that had this disease, then the new cattle would get infected. But Smith and Kilbourne found that ticks were the ones that were transmitting this parasite between cattle. And in understanding that, they quickly came up with a solution, which was simply to let pastures lie fallow for 6 months or more at a time, and then you can move new animals in, because the ticks themselves wouldn't survive more than 6 months. They drop off the animal, lay eggs, and the larvae are looking around for new animals, but they have a finite life, and once that 6 months is gone, the pasture is safe again. So it's been a theme that Andy Spielman sort of instilled in me, which is you use basic biology. You look at questions in basic biology, but those answers to those questions could be used to come up with interventions. And it comes out of this tradition set by Theobald Smith, who interestingly was essentially the founder of our department at Harvard, where Andy Spielman was professor and where I was a student and you were a student and many others. Theobald Smith founded the Department of Comparative Pathology and Tropical Medicine, which later became the Department of Tropical Public Health.
Oh, I always thought it was Tom Weller.
Well, Weller took over. It was called Comparative Pathology and Tropical Medicine, then Tropical Medicine. And Weller took over, and it became Tropical Public Health in the 1950s.
Wow.
And you know, before that, it was actually a medical school department, and then it got more and more over at the School of Public Health, and Weller was a School of Public Health person. Mm-hmm. So these protozoa are common in animals. If you look at the blood of skunks or raccoons around here, for example, they each have their own Babesias, completely unrelated to the one in rodents, which is Babesia Microata. But once upon a time, they were thought to be very host specific. If you found one in a new animal, it was by definition a new species. And all of this got stood on its head when a person without a spleen died of a Babesia infection, which the people in Central Europe ascribed to the European agent of red water, Babesia bovis. And that was considered to be a medical oddity. In fact, classic books on the pathology of infectious diseases at the time showed this to be, you know, just one of these strange infections that we see every once in a while. But in 1966, there was a babesiosis case in California. The identity of that parasite's not known. The slides no longer are available. And all of this has been done because the parasites are readily detected on blood smear using the same methods that you would use for malaria. It's not as sensitive as newer methods, but it's definitive. When you see something like this, you know that this is going to be either Babesia microata or a malaria parasite, or Babesia or a malaria parasite. And the differences between the species, people claim to be able to tell from the morphology of the parasite itself in these blood smears. In 1968, in Ireland, of all places, a farmer died of cattle Babesia divergens, another agent of red water. And then came 50 years ago, 1969 on Nantucket Island, where a woman with a summer home on Nantucket became ill. She had no travel history to a malarious country, but was diagnosed at Rutgers Medical School. She was actually a California resident. And the Rutgers people forwarded the microscope slides to the CDC, and a very sharp-eyed team said, well, look, because the Rutgers people thought it was malaria. And they knew the CDC was tracking malaria cases across the country because it had been eradicated from the US. And the microscopist there said, hey, no, this is not Babesia. This is actually, this is not falciparum malaria. This is Babesia. So it was a very astounding feat of microscopy. I say that because as a graduate student and taking the parasitology class at Harvard, one of the practical exams had a slide like this, and I mistakenly called it falciparum when it in fact was Babesia microti. Excuse me?
A class that you eventually taught, right?
That's correct.
You taught me in that class.
Yeah. And you know, there are some distinguishing features, but if you have a crappy slide or bad staining, it's very easily confused. Mm-hmm. So the odd thing on Nantucket was another 5 or 6 cases were found within the next 5 or 6 years, and only on Nantucket. It was just there, and that prompted the CDC to investigate. But we'll get on that in a minute. In 1991, this dogma that you could only have human infection in splenectomized people with a cattle infection, or in certain sites in the Northeast, infection by Babesia microti. That is, that parasite didn't seem to need immunocompromise as induced by splenectomy. So this WA1 parasite became the sort of the new wave. People realized that, hey, it's not just these cattle parasites and this strange rodent parasite. Other kinds of Babesia may exist. And subsequently, with the attention paid on the West Coast to WA1 Babesia, now called Babesia duncani, Babesiosis was identified in California to a closely related but still undescribed agent there. And then finally, in 1995, yet another parasite was identified in a man from Missouri, again, someone with a splenectomy. So the diversity of parasites that infected man originally was thought to be restricted, and all of a sudden we find out, well, maybe it's a little more diverse than we thought, and they certainly aren't as host-specific as people once said. So the second part of the astounding finding at CDC, the microscopy that they did, was that they actually assigned it to Babesia microata, knowing that an infection called Smithia Microata had been described from mice in Portugal, and in fact people had used it as a laboratory plaything because it was supposed to be harmless to humans. And so you had this woman appear in subsequently several cases on Nantucket, and then nearby Martha's Vineyard, Cape Cod, and then on down the terminal moraine islands of Block Island and Long Island. All of these places finally started identifying cases of babesiosis. The malaria surveillance team sent somebody out to CDC. And they isolated the parasite, and they investigated this cluster of infections on Nantucket. and as we talked about in the preceding conversation, Andy Spielman identified the white-footed mouse as the main culprit in infecting ticks, and also that there was a new species of tick that was transmitting it between mice and to people. The 59-year-old woman who got babesiosis removed a partially fed tick from her shoulder girdle a couple weeks before getting fever. And so because it had been assigned to Babesia very quickly, it didn't require any thought at all. All Babesias are maintained by ticks and other animals. It meant that those cases on Nantucket had to be acquired from tick bite, which meant that Spielman could focus on ticks as the most important thing to understand. Now we know that this diversity, once upon a time, it was very immunocompromised people lacking a spleen or rodent Babesia, and then the floodgates opened. And now we sort of recognize two epidemiological patterns. We have this endemic babesiosis or Babesia microta babesiosis, which exists not only in the United States, mostly in the northeastern and upper Midwestern U.S., but also across Europe. There have been 2 or 3 cases found in Europe. It certainly is, the parasite is in rodents all the way from Alaska into Japan. And one of the big questions that remains to be answered is why on earth these parasites that are in rodents, which are genetically very, very similar to the one that is on Nantucket, why they don't seem to cause disease in humans. Some of this is related to the fact that the nymphal stage of the tick vector in Europe, the sheep tick, doesn't seem to be involved in maintenance of Babesia there, but rather a very narrowly feeding tick, Ixodes trianguliceps, may actually be the one maintaining it between rodents. In Russia, Ixodes prissulcatus, all of these are very closely related. In fact, if you looked at all of them simultaneously, you would have a hard time telling the difference between the American deer ticks, the sheep tick, and the taiga tick in Russia. But anyway, you have this possibility for endemic zoonotic babesiosis due to Babesia microta across a very wide range of countries. Then we have what we call sporadic babesiosis, where you can see isolated cases or maybe a few cases a year in almost any country. Most of these are due to cattle parasites caused by Babesia that cause disease, Babesia divergens, Babesia bovis, Babesia EU1, Babesia capreoli. And then you have other ruminant Babesias, things of domestic animal production concern for sheep or goats found in China and Korea. And then you have ones where we really don't understand what's going on, but the evidence is fairly decent in terms of presentation of good photographs of microscope blood smears of parasites in people. And then finally, there's some suggestion in other countries which are less well documented. So the thought is Babesia actually does occur across the world and is just understudied in most places. And there's a reason for that. In most of the tropical areas, by far the more important infection is malaria. Malaria, some people have speculated, oh, well, a lot of malaria is diagnosed on blood smear. Maybe they're just confusing Babesia with malaria with Babesia. That remains to be seen, but I think by now, with all the molecular tools that we have for diagnosing protozoan infections, people would have found more Babesia if it was much more common than we think. So how do people get Babesia? What are the characteristics of people getting it? The cases for babesiosis, and this includes the odd ones, the deer-maintained ones, such as the WA1 or Babesia duncani and the California type Babesias. They're mainly localized, again, where you see Lyme disease, the Northeast and upper Midwest. There's no real pattern on the West Coast. But certainly Babesia microta is the most common and the one with the most public health concern. We're lucky to see 1 or 2 cases of Babesia duncani every 5 years, at least bona fide, well-documented Babesia duncani. But there are hundreds of cases of babesiosis reported each year in the eastern United States due to Babesia microta. Nonetheless, and you and I spoke about this a little bit, babesiosis still seems to have more of a restricted distribution than Lyme disease. Not every place with Lyme disease has Babesia microta, but every place with Babesia microta has Lyme disease, as at least the ones that infect humans. There are strains of Babesia microta. Maybe they're slightly, maybe they deserve their own species name, such as those in Alaska, where there is no Lyme disease. But at least in terms of human public health problems, where you see Babesia, you certainly see Lyme. But where you see Lyme, you don't necessarily see Babesia. And neither infection track the distribution of their vector, although there is, at least in my mind, there's controversy as to whether it's one and the same vector. Today and for the last 20 years, people have lumped all of the ticks in the eastern United States under one name, Ixodes scapularis, and rejected the name Ixodes damani for the human-biting ticks of the Northeast. There is no difference in the laboratory in their capacity to transmit, but it is interesting that populations from the south of this tick do not seem to attack humans as nymphs. And I argue that deserves more scrutiny than people have put into it. And I continue to irritate people by using the old name Ixodes damani whenever I can. You yourself and your lab, and most recently Guang, published a really nice paper that should really get people thinking. But no, that it's settled science. Let's move along. Nothing to see here, folks.
Yeah. The most telling thing about that is if you go back and try to find the type specimens from 1823, well, first of all, they don't exist. And if you find subsequently, I think it was Nuttall and Warburton said that they were useless descriptions anyway. So some ways.
Yeah, no, the nomenclature is really all messed up. Although Kieran's designated a neotype for scapularis, and so that has to replace it. But nonetheless, the hypothesis to be rejected is that this is a species complex, and it's not as simplistic as the original paper sinking the northern deer tick name Ixodes damani make it out to be. I'm still puzzled as to why people don't reopen the investigation, especially now that people have realized that in St. Petersburg and in Russia, you have hybrids of Ixodes pavlovskii and Persilcatus that form fertile hybrids, F1-5 hybrids, and that they are even more vector competent than either of the parental species. And so this idea that, oh, well, we demonstrated conspecificity by showing that we showed that the F1s are fertile. It means that they're the same species. That is just simplistic high school biology understanding of what a species is. Yeah. But don't get me going on that. The cases of Lyme disease appear to have been rising. They have risen. This is probably real here, but in here, some of it is due to the fact that there was better reporting, that it was more on clinicians' minds. But it certainly has been going up and up and up due to the expansion of ticks across the eastern U.S. and the upper Midwestern U.S. And in Massachusetts, you see the same thing with better reporting. You started seeing more and more cases, and it's probably on the order of 3,000 to maybe 30,000 cases a year, even in a state like Massachusetts. A lot of cases are not reported at all. They simply appear as a rash. With babesiosis, because a lot of the diagnosis depends upon seeing it in a blood smear or now with polymerase chain reaction evidence. Those are laboratory tests, and all laboratory tests are electronically submitted to the Department of Public Health. And so I think we're getting a better handle on how much babesiosis is out there. Babesiosis, and we'll talk about this in a minute, is largely asymptomatic in most of the patients that get it. But there is obviously an increase as well. The tick has increased tremendously in Massachusetts ever since I've been working on it. Once upon a time, we said, you're never going to find deer ticks unless you're 30 kilometers from the coast. Everything inland is just too cold. And now we see them everywhere. There's no place in Massachusetts where you don't see have deer ticks. Mm-hmm. So there is an element to both, that is increased reporting, but also a transport and establishment of new infestations of ticks, probably by birds. There are more and more receptive sites. I just came across a reference recently from Mass Audubon that says 40 acres of land is manipulated every day for new developments in Massachusetts, which is just tremendous. And most of that land is forested, which means that you're creating the conditions that probably established this infection in the first place. That is, you clear cut the forest, you let it come back in successional growth, and the animals that exploit that successional growth are generalists such as deer and white-footed mice, and the ticks like the successional growth because it's nice and moist, and your houses are right up into it. Mm-hmm. But when you see graphs like this, you always have to think it's not just about the tick, it's also about us, that we recognize it and report it better. Mm-hmm. Not only on top of all of that, there's clear changes in the parasite itself. Natural selection operates every single day in natural populations. We've suggested that there's been a expansion of a lineage. There are at least 3, maybe 4 different lineages of Babesiosis Microata in New England. And when I say lineage, it means that they're distinguishable by genetic tests, and there may be associated phenotypic changes. That is, maybe one of these lines is more easily transmitted by ticks. Maybe there's more of a parasitemia. Who knows? But it appears that New England has been taken over by one lineage coming from the south and expanding northwards. And that we had originally thought when we started this work was that everything would radiate out of the terminal moraine islands, and particularly Nantucket, and that's how the mainland got seeded. That's not true. There are a number of different lineages that we find across New England, and they have been in existence in parallel with Nantucket, but they're now expanding, whereas Nantucket being isolated is not contributing to any of this. And so this may be associated with increased risk to humans as well. And that's an ongoing research. What is this like for people? What is babesiosis for people? Just as Theobald Smith found with cattle redwater, with Texas cattle fever, the older an animal is, the older a human is, the more likely it is that there's a severe disease. However, Lyme disease and anaplasma, both of them seem to affect people across the age distribution, whereas those with babesiosis are much more likely to be reported as cases if they're older. Actually, anaplasma also is age-related, but Lyme disease occurs across the whole spectrum, other than here, which we don't understand at all. The age cohort from 15 to 34, you would think is the most outdoors active cohort out there. The people in the prime of their life being exposed constantly, yet they're less likely to be reported as cases of Lyme disease. And that's something that's a really interesting research question that really isn't being approached by anyone. It could just be, they're just really healthy and they're living life right, and they were able to fight off Lyme disease, as they certainly do for Babesia and Anaplasma. They're fighting off infection, even though they're undoubtedly exposed. And this trend is seen nationally. Most of the cases are occurring in people who are older than 50 years old. And fewer and fewer from younger groups. What's it like to have babesiosis? After getting bitten by a tick, symptoms and signs could appear as early as a week afterwards. In experimental animals, I don't see parasites in the blood until at least 3 days after the ticks have finished feeding. It's possible that you might detect them with more sensitive methods, but I point out that if you can't detect it by blood smear, it's unlikely it's going to be causing much pathology, any symptoms and signs. People report just feeling awful. They may be off their food. They may not want to eat. They're fatigued. And then the hallmark of babesiosis is fever. Very few infections. Tularemia and spotted fever are the only other ones that will produce a fever of 103, 104, 105. And so that's what really brings them to the attention of a healthcare provider. Along with the fever, you have episodes of drenching sweats and chills and muscle aches. And this is due to the liberation of cytokines and other pro-inflammatory substances by the release of parasites as they exit the red blood cells that they've lysed. They deplete the platelets, so you have what's known as thrombocytopenia. You have fewer red blood cells as the infection progresses, and the white cells are down. The liver enzymes become elevated, and all of this depends on parasitemia. The splenectomized people, the spleen is extremely important in clearing parasitized red blood cells. And it's thought that's probably the main reason people with splenectomies are at high risk of severe disease, where there have been case reports of people with 85% of their red blood cells containing parasites, splenectomized individuals. And if you can't control that, that doesn't leave you an awful lot of oxygen transport in the body. Immunocompromised people with HIV, people who are on drugs that suppress B cell production, people with leukemias, for example, they're also at high risk for high parasitemias. And that shows the influence, particularly of B cells, which means that antibody is important in controlling this infection. And most people don't appreciate, even with treatment, 5% of the cases can die. So this puts it right up there with things that have much more of a severe reputation, such as tularemia, where the case fatality rate's actually 5 to 7%. So this is not this of all the infections transmitted by deer ticks, other than Powassan virus, this is the one where there can be complications that can terminate poorly, especially if you're older or immunocompromised. The treatment is not all that great. But I did mention that most people who get it are not hospitalized. They have many young people are just completely asymptomatic. I had mentioned premonition, this concomitant immunity, where the parasite stimulates antibodies to be produced. The antibodies in turn control the parasites and keep them at a low enough though, which is continuing to make your immune cells produce antibody. So that's probably why there's a lot of asymptomatic infection. This is what characterizes infection in animals. In humans, most people are not hospitalized, and a minor fraction are hospitalized as many as 10 days. So in general, people do all right with babesiosis. However, the asymptomatic infection, it used to be we would concentrate our efforts on trying to understand severe babesiosis. We still should do so, but because healthy people may be infected and not even know it, it's become the most commonly reported transfusion-transmitted protozoal infection, and in fact, one of the most common infections, period, in the blood supply. Screening is not done everywhere, and even though this doesn't seem to be a lot, 0.4% of 1.5 million donations, that's sufficient so that it's, there have been well over 300 transfusion-transmitted cases recognized. And when you consider that the people who receive transfusions are generally those who are not doing too well to begin with, it becomes a problem. And so there have been a lot of calls for actually adding screening for babesiosis to the blood supply. FDA recently came out within the last 2 years a recommendation for targeted screening. And the interesting thing here is that even though we would expect to see most donations come from the middle of the summer when Babesia is being actively transmitted by ticks, we actually see transfusion-related cases and donations, positive donations from every month of the year, which means that people who are healthy may have parasites circulating in them for months and months and months. So this was a landmark study done by the Red Cross where they looked at, 1.5 million donations and came up with the fact that, a good number actually contained parasite DNA, and that was inferred to be sufficient cause to remove, right now any unit of blood that screens antibody positive is removed from further use. So that was really everybody thought asymptomatic infection. So what? We don't even want to treat you. Now I think that's changed. If they identify someone, they will be deferred from blood donation as well as be very likely be treated. When do you get babesiosis? Well, like any of the other deer tick transmitted infections, the bulk of the cases appear in June, July, and August. This is when the nymphal stage of the tick is most common. You have right now in the fall, you have adult ticks out. They'll be out throughout the cold months until the end of May. If they feed now or in the spring on something large like a deer, they'll lay 2,000 eggs. Those eggs will hatch synchronously. Doesn't matter if the tick fed now or if fed in the spring. They all hatch in July, and the larvae start looking for animals in July, the baby ticks. The larvae are feeding on something small like a mouse or a bird or a shrew. If the mouse or shrew is infected with Babesia microata, the larvae pick it up, and then they will overwinter. The larvae are feeding July, August, September. They will overwinter, and as it warms up in the spring, will turn into nymphs. The nymphs then, the larvae are uninfected. If they pick it up, the nymph is the stage that will be transmitting. The nymph is around May, June, and July, which coincides with the peak of transmission for Babesia as well as Lyme and the other tick-borne infections that we see. This is actually anaplasma. Babesia here, Borrelia miyamotoi, Lyme disease, and anaplasma. And Babesia is, as you noted, the prevalence of infection in host-seeking ticks is about 1/3 as much as that for Lyme disease. For Lyme disease spirochetes, so you can have 10 to 30% of the nymphs infected, 5 to 15% of the ticks are infected by Babesia. I think a lot of emphasis, too much emphasis is placed on these numbers, infection rates, mainly because we don't see, look here, you don't see cases of Lyme disease to any great extent in the fall or in the winter time or in the early spring when the adult ticks are around, yet 40 to 70% of the ticks are infected. And so it means that the most important aspect is clothing, tick size, and removal and inoculum. That is, people finding ticks and removing them in time. But remember also, when it gets colder, we dress differently, and ticks have a harder time getting up underneath the clothing. So human behavior modulates apparent what we call entomologic risk. Nonetheless, this is evidence that the transmission cycle is much more fragile than it is for Lyme disease spirochetes. I do want to briefly mention the basis for promptly removing ticks. This idea of a grace period. Most people say we don't believe this happens. You can get infected the moment a tick feeds on you. It's just not true. There's a biological basis for this. Remember, larvae turn into nymphs over the course of 8 months. The larvae feed August and September, and they start looking for hosts in May and June. 8 or 9 months. If you were a microbial agent stuck within the tick for 8 months and you're continuously replicating, you need resources in order to replicate. That means you're depleting your host resources, which means that it's affecting the fitness of your host. So over time, there's been some sort of adaptation or agreement between the two, the host and the parasite, and the parasite becomes dormant within the tick during those cold months. And they're non-infectious when the tick is out looking for animals, and they get infectious when the tick, which is its body temperature is whatever is ambient. So it's, let's say, 10 degrees centigrade. When it achieves a host, at least a warm-blooded host, that change in temperature from 10 to 37 degrees centigrade says, wake up, it's time to get out. The blood coming in serves as a nutrient, and the organism changes. With the case of Lyme disease, you have a change in the outer surface coat from an outer surface protein A to an outer surface protein C. And this takes 24 to 48 hours to happen. But there's some superb molecular biology showing gene regulation, changes in the regulation of genes that control the expression of these proteins during this time. With Babesia, it's even more prolonged, that it takes 48 to 62 hours before the parasites within a tick become infectious. So a tick has to be on 48 to 62 hours before there's any kind of infection possible, because they in a flat tick, in a tick that's out there looking for hosts, the parasite is in this undifferentiated form. And during the course of 48 to 62 hours, it splits up into thousands of infective particles. It's only when those infective particles are available that you can actually cause infection in whatever the tick is attached to. Same thing seems to happen with anaplasma, but sadly with tick-borne encephalitis viruses such as Powassan virus, there is none of this dormancy and reactivation. But I always like to try to dispel the idea that you can get infected right away from a tick bite. There's no such thing as all or none phenomena in biology. It's not that it doesn't happen, but in the original experiments with Lyme, the guy who was doing the work, Joe Heisman, sat up around the clock doing this. 1 out of 10 hamsters with infected ticks became infected when the ticks were removed before 24 hours. 5 out of 10 became infected, 2 out of 10 became infected at 36 hours, 5 out of 10 at 48, and all 10 out of 10 at 72 hours, noting that ticks take several days to feed. They're unlike mosquitoes. Day one, they're preparing the site of the bite. They're spitting stuff into you to cause a pool to form. And then on the last day, they swell up 20 to 100, 20 to 50 times their original size. And so all ticks take a long time to feed, and the pathogens within them are adapted to this long time.
Just a friendly amendment to that point, because I know there are people that will be watching this that are sitting up now in their chairs and taking some concern about this. So one thing I think about is that a plausible explanation for why people have a lived experience where they were bitten by a tick and they know that tick was on for them only for a few hours because they see that part of their skin. The tick wasn't there 6 hours ago. Now it's there. And so they probably are correct that tick is not fed for a long time. But when they try to close the circle between that tick and their diagnosis with Lyme disease or Babesia, they disregard the fact that a second tick that would bit them over here that was on for a length of time necessary, and when undetected, gets forgotten in the piece. So there's that piece of it. And the other part of it is just to remind people that these are biological phenomenon, so they are distributions. So
that's correct
about 48 hours or 24 hours, that there's some things that might be closer to one end of the tail or the other. So
you're absolutely right. But trying to get that concept across of probability of events is not easy with even our colleagues. Actually, there is one instance where you can have very rapid transmission, and that is when you have an animal such as a cat, an outdoors cat, indoor outdoors cat, which has a tick on it for 36 or 48 hours, comes in, grooms itself, the tick falls off and finds that person and attaches, then you can have transmission fairly instantaneously. So that certainly happens. And in fact, we have a paper that was just accepted showing that ticks from vegetation, a larger proportion than we expected, actually have evidence of multiple blood meals, so that they feed, get indigestion or get groomed off and find another host. So it happens more frequently than we think.
Mm-hmm.
So that's really all I want to say about babesiosis. I hope I've covered the relevant points for at least people understanding when they can get infected, what they should look for in terms of signs and symptoms, and some of the broader ramifications such as transfusion infection.
Yeah, another point maybe, and maybe you made it and I missed this, but the treatment. So the notion that there's a prophylaxis or a quick treatment with doxycycline that may be effective against prevention of disseminating Borrelia infection. Or what are the consequences for Babesia in that circumstance?
Yeah, that's a very important point, because in some places, like I can swear on Martha's Vineyard in Nantucket, we've started to see a decline in Lyme disease cases, but babesiosis remains the same, if not more. And I'm starting to wonder, I've worked very hard to get the healthcare providers out there to just pass the doxycycline out after a recognized tick bite. That anyone with a tick bite, no questions asked, here's your 1 pill of doxycycline. It's probably impacting Lyme transmission, but of course it does absolutely nothing for Babesia. And so these are the sorts of things that epidemiologists have to keep in mind when they're analyzing data. It's sort of like, well, there's something interesting going on with Lyme. Oh, well, maybe it's just this intervention, this simple intervention. We don't have evidence as to whether it works against Borrelia miyamotoi or anaplasma, the 1 tablet doxycycline. But we do know that doxycycline is the treatment of choice for both, and it wouldn't surprise me if there is some efficacy, but there's certainly no efficacy for Babesia microta, and certainly no efficacy for Powassan virus.
Any prescribed prophylaxis for Babesia?
Well, sadly, we won't see a vaccine. It is a close relative of malaria, and we all know how long it's taken for them to even have clinical trials of a vaccine, which is moderately effective. It may modify disease, but certainly not in the efficacy range of typical vaccines. But the most important issue is to prevent tick bites. It's going to be forever before we have vaccines for every specific microbial agent transmitted by ticks. We'll have a Lyme disease vaccine in 4 years. I think that's pretty certain. But that doesn't mean people shouldn't be preventing tick bites to avoid infection by Babesia, by anaplasma, or by Powassan. So it means that we always have to prevent tick bites. And I've been pounding on, whenever I lecture to communities, it's like, well, if you have kids and you want to send them out or let them go ride their bicycle, you say you got to wear a helmet. So I'm going, if you have kids and you live in a tick endemic site, and today, most places are pretty tick endemic, have them reach for the repellent, DEET, the Keratin, the best thing in the world the single most useful thing people can be doing, treat their clothes with permethrin. Permethrin is extremely safe for humans. Military uniforms have been treated with it for 30 years now. Our military, they're in those uniforms 24/7. The permethrin is the active ingredient in Nix, which is the standard treatment for head louse. FDA allows a 1% solution of permethrin to be applied to a child's head for 30 minutes. And permethrin treatment of clothes is at most 0.5% permethrin applied to clothing, and it's bound very tightly to fabric. So it's safe, and it's death on ticks. I can't have permethrin anywhere near my car or myself.
Because you want
so prevention is key. Absolutely.
Yeah. Well, that's great. That's a great note. That's a great note to end on. And thank you very much for doing this.
No, you're doing a good thing. And so you say the audience is, the people who are out there in the bushes.
That's right. Yeah,
so that's great. Yeah. Yeah. And, today with the way the media is disseminated, it's undoubtedly going to get more hits than just your intended audience. At least if people think about it.
Right.
Well, good to see you, Steve.
Good to see you. Thanks so much.
Yeah, anytime.
Take care.
All right.
Bye-bye.